Meiosis 1. Meiosis 1 has 4 stages: prophase, metaphase, anaphase, telophase. The first stage is prophase, and what the differences and meiosis begin to appear, such as chromosomes begin to condense, which is right here. These are the chromosomes, and as you can see, they're starting to get smaller and closer together. The DNA is broken at the same spot on each homologue. So as you can see, on each part of the chromosome, they are broken, which means they're like split up here, and they're also split up around there. Metaphase 1. Homologue pairs, not individual chromosomes, such as these, are going to be set up into pairs. So like homologue pair, homologue pair, homologue pair, homologue pair. How the lines are set up is random. So basically, you see how this is pink pink, and then that's pink purple, and then this is purple purple, and then this is pink or purple pink. That's random, and it can happen in any way. And the orientation equally able to have been flipped. So this side could have been over here and this over here, basically. And then you could also have taken it like this and brought it upside down, so that way it would have done the same thing. But this would have been on top, this would have been on bottom, this would have been on top, that would have been on bottom.
After metaphase comes anaphase, and if you remember how metaphase looked like they were together, in here they're pulled apart. So the homologues are pulled apart. So as you can see, they're now separate, pulled apart to opposite ends of the cell. And the sister chromatids, which are these two, not all of them, just this one in general, stay together. And as you can see, it's purple, pink, because when they were fused, they took some from each other. And after anaphase comes telophase. Chromosomes arrive at the opposite end of the cell pole, and in some organisms, the nuclear membrane reforms chromosomes, and sometimes the cells skip this part, but not all. So as you can see, they all went to opposite ends, and the cell begins to split up to then go into meiosis 2.
Meiosis 2. The cells made in meiosis 2 are haploid ones that came out of meiosis 1. Meiosis 2 is a much simpler and more straightforward, shorter attempt and process than meiosis 1 is. In prophase 2, from meiosis 2, chromosomes condense and the nuclear envelope breaks down. So this is a nuclear envelope right here that breaks down, and these chromosomes condense right here. And then in metaphase 2, as part of meiosis 2, the chromosomes get into a line right here, and then along the metaphase plate, which is right here, because after the spindle has been formed from prophase 1 or prophase 2, then it's like a plate right here, because you see the spindle right here with these different lines and the two top and the bottom, and that's how it goes.
So after metaphase 2 comes anaphase 2. And in anaphase 2, the sister chromatids separate and are pulled towards the opposite poles of a cell. So as you can see, back to metaphase 1, these were they were together, and this is when they're apart. After anaphase comes telophase, and it's when the nuclear membrane form around each set of chromosomes and decondenses to split into new cells.